So I would like to start my presentation with talking a little bit about deep Gestalt, because this is the technological basis of the analysis that we did. So deep Gestalt is a deep convolutional neural network that has been trained using thousands of photos of children that are affected by different genetic disorders. When we are presented with a new input photo, first, we do some pre-processing. First, we detect the face. We also detect the facial landmarks that you see here in green. We align the face first, and then we crop it into different regions. You can see here, for example, the eye area, the mouth area, and more. Then each of these regions pass through the layers of the neural network. And as you can see here, with each new layer, we are basically extracting the most relevant features from the face that represent the facial phenotype of that child. Then, in the end, we aggregate this information from these different regions, and we get a vector with the top 30 most likely syndromes that the child might have, which are basically the most similar to the facial phenotype of that child. Now, if we look at the final layer before the syndrome classification output in Deep Gestalt, we actually have this vector of 320 values, basically this vector of numbers, that is a numerical representation of the facial phenotype. Now, if we condense these high dimensional vectors into just two values, 2 dimensions, we are then able to represent each face in a 2D scatter plot like you see here, where each data point represents a patient's photo. Now, the smaller the distances between these data points, the more similar their facial phenotypic features are. You can see here, for example, that patients that have been diagnosed with the same syndrome, for example, here Coffin series, tend to cluster together in this space. Now, the name of this technology is GestaltMatcher, and we can leverage these facial phenotype descriptors, these like data points, these vectors, for syndrome classification, delineating new phenotypes, and also clustering patients. And this was exactly the technology that we use for the analysis for the publication that we are discussing here today. So we took the photos from the selected patients that Erin just presented, and we also took age, gender, and ethnicity matched photos of our target comparison groups. These are the unaffected controls, here in blue, represented in blue, Smith-Lemli-Opitz in orange, and fetal alcohol syndrome in green. These two are potential confounder syndromes, as Erin previously covered. We then converted each photo from these groups into their facial phenotype descriptor, basically the data points that we are seeing here in this plot. And you can also see these ellipses that are represented here. And they are actually one standard deviation around the cluster center. So you can think of them as the cluster core for each of these groups. And we can already see, just from looking at this visualization, that there is already some separation between the potential novel syndrome here in red and the other groups. If, for example, we represent these same high dimensional vectors, but now in 3 dimensions, meaning that we are basically condensing a little bit less the information, right? We are able to visualize a little bit more data here. We can see that the red crosses here that represent a novel syndrome still separate even maybe a little bit more clear than what we saw in the previous 2D plot from the other comparison groups that we are analyzing. Now, the question that we wanted to answer is if the novel syndrome is more similar to itself than to the target matched groups. To answer it, we first compare the novel syndrome to itself with what we call an intragroup similarity analysis. What this means is we take, for example, this data point from the potential novel syndrome, and we calculated the cosine similarity with basically every other point within the novel syndrome. Then we repeated this process for the second data point, calculating the similarities that were not previously covered, and so on with the remaining data points. So this is the intragroup similarity, where we are comparing the novel syndrome to itself. Then we also wanted to compare the novel syndrome to the other groups with intergroup similarity. So we did the same cosine similarity comparison, but now we are comparing all the data points from the novel syndrome with all the data points from each of the other groups. For example, here between the novel syndrome and the unaffected group, here with the Smith-Lemley-Opitz group in orange, and also the fetal alcohol syndrome group. Here you can see the similarity distribution between the novel syndrome versus itself, and then the novel syndrome versus the other groups that we have now discussed. If you are not familiar with this box plot visualization, you can think of it as this top-down view of the distribution. We have the minimum and maximum values, we have the median value here in the middle, and the first and third quartile. So in this visualization, we can already see that there is at least a tendency for the similarity of the new syndrome compared to itself being higher than the new syndrome compared to the other groups. But then we wanted to conduct some statistical tests to be sure that this was really happening in a significant way. So prior to that group comparison, we made sure that we could apply parametric testing. So the samples that you see here are independent. They have normal distributions, equal variance, and also no outliers. So then we conducted a one-way ANOVA to compare the 4 groups. This test compares the means between the groups and determines whether any of those means are significantly different from the others. As you can see, our results had a p-value that is well below 0.05, meaning there is a high statistically significant difference between these 4 groups. But however, the ANOVA does not really tell us between which pairs of groups this difference occurs. So we needed to follow it with independent t-tests between the new syndrome and the other groups. So that's exactly what we did. And based on these results, we can say that the novel syndrome intragroup similarity was indeed significantly different, and as we saw before in the other plot, higher than the similarity with the other groups. So these findings suggest that the novel syndrome has a facial phenotype associated with it that is cohesive and separates well from the phenotypes of the other syndromes and also the unaffected groups. So if you are interested in conducting this type of research and analysis using GestaltMatcher or Deep Gestalt, as I introduced before, feel free to contact us at research@fdna.com, and we'd be very happy to support you with your research projects.